Welcome to a screencast on the Determining an Equilibrium Constant lab. This screencast will go over the theory involved in the lab and will give you some hints on how to do the calculations. So let's get started. First of all, the reaction that you will be determining an equilibrium constant for is the reaction between the iron 3 plus ion and the thiocyanate ion to make the complex ion thiocyanoiron 3. Now, the iron 3 plus and the thiocyanate ions are both clear and colorless, and these are in aqueous solution, as is the product thiocyanoiron 3, and that complex ion is a reddish-orange color. And that color is important because we're going to use that color to determine the concentration of this ion, and then that'll be used to determine the concentration of the other ions as well, at equilibrium, of course, and that will then allow us to determine the equilibrium constant. And of course, the equilibrium constant for this reaction, it's going to be a K sub C because we're doing this in terms of concentrations of the solutions, and it will be the ratio of the concentration of the FeSCN2 plus ion at equilibrium over the product of the concentration of Fe3 plus times the concentration of SCN minus ions also at equilibrium. And that will turn out to be a number. And you'll do this experiment, you'll do several trials of it, and you will get roughly the same number for each of the trials. You'll average those numbers, and that will be your reported K sub C value. And then it, of course, needs to be reported for a specific reaction and at a specific temperature, and this reaction will be done at room temperature or 25 degrees Celsius. Okay, now here's how this works. You're going to use, well, actually, you're not going to use, this will be done for you and you'll observe the results, but a spectrophotometer is used to measure something called the absorbance or the transmittance of a colored solution. So the solution we'll be using will be sort of an orange-red. That will have some light that shines through it. That light is a complementary color to the orange. The reason this solution appears reddish-orange is because its complementary color, blue, is absorbed. So pay attention to the specific wavelength used. We use a wavelength of light where there's a high absorbance in that solution. And the guts of the spectrophotometer are that there's the light source that emits light at a specific wavelength, and then it goes through a lens and a prism or a grating and then a slit to make kind of a beam of light. And there is a certain intensity or amount of light that is coming into our sample. The container that we use for our sample in a spectrophotometer, it's kind of like a little test tube, but we call it a cuvette. And so light will go into that. Some of it will be absorbed, some of it will be transmitted, and then there's a photocell or a detector that measures the amount of light that's transmitted compared to the light that is absorbed. It'll actually give out a percent transmittance, and then we will actually convert that into an absorbance, where the absorbance A is equal to the negative log base 10 of the transmittance, and we are going to use absorbance because it turns out that that is what's proportional to the concentration of the solution. So as the solution, when we have a solution that has a higher concentration, it'll be a darker orange-red, it'll absorb more light. When we have a lower concentration solution in that, and by concentration this is of course in terms of that red-orange FeSCN2 plus ion, if there's a not as concentrated, a more dilute solution, it'll be a lighter color and it'll absorb less light. And it turns out the absorbance is directly proportional to the concentration of the colored species in the solution. In this case, this is called Beer's law. You may have, probably have, run across this before. And the lowercase a and lowercase b are constants that depend both upon what's called the path length, so how big your cuvette is, how wide a path the light goes through that contains colored solution, and then there's also a constant that depends upon the specific solution that you're dealing with. And those don't really matter for us right now. What matters is that the absorbance is directly proportional to the concentration, and that the absorbance is determined by taking the negative log base 10 of the transmittance. Quick note, the meter will display percent transmittance, and you take negative log of the transmittance as a decimal. So, for example, if the transmittance turns out to be 75%, you will take negative log base 10 of 0.75. Okay, so this will be done for several solutions in what's called making a calibration curve. So there will be solutions prepared to make a calibration graph. They'll all involve the same reaction. And what's going to happen in this case is there will be a certain amount of a 0.200 molar solution of the Fe3 plus that gets combined with a certain amount of the 0.00200 molar SCN minus solution. When those are combined, they'll react and they will make the FeSCN2 plus ion. But a couple of things to notice. This is just a sample calculation. You'll have to use the correct numbers given in the experimental data. But let's just say we have 50 milliliters of this 0.2 molar Fe3 plus solution, and we have, say, 5 milliliters of the 0.002 molar SCN minus solution. Now notice the Fe3 plus has a much higher concentration and a much larger volume, so it's in a huge excess compared to the SCN minus, which has a much lower concentration and a much lower volume. And what that means is the reaction will use up all the SCN minus. It's essentially, well, it is a limiting reactant. There will still be a whole bunch of Fe3 plus left over, and that drives the reaction just about to completion in terms of forming the FeSCN2 plus ion. And this is done on purpose because it then allows us to determine the concentration of the FeSCN2 plus ion that was formed. So what will happen is when these are combined, they'll make an orange solution. They'll be combined in a 100 milliliter volumetric flask, so they end up getting diluted as well because some water is added. And then each of these samples will go into the colorimeter and we'll get its absorbance, but we'll now have to note we can calculate the actual concentration of the FeSCN2 plus in this solution by noting that we took 5 milliliters in this example of the thing that we're going to run out of, the limiting reactant, SCN minus. We're going to dilute it to 100 milliliters total. So this is our dilution factor. We dilute by a factor of 5 to 100, and that's going to, of course, dilute the concentration of everything, of what we started with and the concentration of the solution we made. So that dilution factor then gets multiplied by the concentration of the solution that gets used up, because this will limit how high a concentration of the solution we form, or how high a concentration of this solution that we form will be. And so this then gives us a concentration of, in this example, 0.000100 molar for the concentration of the FeSCN2 plus that is formed. And you will do, or there will be, I think, 6 samples that are done with differing amounts of the Fe3 plus and the SCN minus. Each one of these ends up getting diluted and completely reacting to form FeSCN2 plus, and so each one of them will have a different concentration of FeSCN2 plus once the reaction's taken place. Okay. And of course, I should note that that solution will have its absorbance measured, and so you'll also get absorbance values along with your calculated values of FeSCN2 plus, which we just talked about. All right, so we now are going to remember that absorbance is directly proportional to concentration. So if we make a graph of absorbance versus concentration, and we'll do concentration on the x-axis and absorbance on the y-axis, and we will plot our data points carefully. And if you do it right, you should get a pretty straight line. And that straight line will have a slope, an equation of y equals mx plus b, where m is the slope and b is the y-intercept. And of course, for us, x is the concentration of FeSCN2+, and y is the absorbance. So this will really be A equals m times c plus b. And you'll get the equation of the line with the slope and with the y-intercept. And what that's going to mean then is, if you do subsequent experiments, and this will be the next part of the lab, if you measure its absorbance, you can then determine its concentration by using the equation, plugging in absorbance, you know the slope, you know the y-intercept, solve for concentration, and that will then give you the concentration of the FeSCN2+ in the new solution you made. Okay, so now we do how we're going to determine the equilibrium constant. And again, here's the reminder of the equation. And we'll once again combine some Fe3+ with some SCN- solution. But notice this time, both of them will have about the same, well, in fact, exactly the same concentration, 0.00200 molar. So neither one is in huge excess. They're relatively the same as each other. And so what will happen then is the reaction will take place, but it won't go completely to make FeSCN2+. You'll reach some position of equilibrium with some FeSCN2+ that's been formed and some Fe3+ and SCN- that are left. So we'll combine these, we'll mix them together and make, I believe it'll be 10 milliliters of solution. So there'll be some dilution there as well. To make a total solution volume. And then we will do kind of the same thing, which is measure that solution's absorbance. And then using the calibration curve we just talked about, you'll be able to determine the solution concentration. Now, once again, we have to remember we have dilution taking place. So I'll just do this example for one of the solutions. You'll have to do it for both of them. So let's say that we have 5 milliliters of the 0.002 molar Fe3+, but then we combine that with 3 milliliters of the SCN- and then 2 milliliters of water to make a total of 10 milliliters of solution. Well, we have a dilution now by a factor of 5/10. And that is of our original 0.002 molar solution of Fe3+. That gives us a 0.00100 molar solution of Fe3+. And this will be what our initial concentration of this is. And kind of a quick note, essentially what we're doing is we're saying, let's pretend we could somehow mix these two solutions together and somehow stop the reaction from taking place temporarily, so we can just take into account how much they got diluted when they're mixed together, so we can get the real initial concentration, because it isn't really 0.002 molar. Everything gets diluted. So the real solution concentration for Fe3+ initially is, in this example, 0.001 molar, and then it'll react and its concentration will go down as it makes reacts with SCN- to make FeSCN2+. That'll be a thing we do in the next step, and we'll use an ice table. Okay, so again, note this is just an example calculation. You'll have to do the calculations for your data, and then you'll have to do a similar calculation for SCN-, but it's the same principle. Okay, now we're almost there. Here's the last thing. Now we're ready to determine the equilibrium constant, and we will do this by using the data collected and an ice table. And so our initial concentration of FeSCN and Fe3+ and SCN- will be the things we determined here from this dilution. So for one of them, it's going to be Fe3 plus has a certain concentration, and SCN minus has a certain concentration, and they won't necessarily be the same. And again, our initial is we imagine we could somehow mix them together and they hadn't yet reacted. And so we don't have any FeSCN2 plus that's been formed yet. Now, of course, what will happen is then the reaction does in fact take place. The change will be that Fe3 plus goes down by some amount. SCN minus goes down by the same amount because it's a one-to-one stoichiometry. They're being used up, so it's negative. And then FeSCN2 plus goes up by the same amount. Again, it's the same. It's a one-to-one-to-one stoichiometry, but this will go up. We don't know what X is yet, but we'll get there. And then at equilibrium, we'll have our initial minus X for the Fe3 plus concentration, our initial minus X for the SCN minus concentration, and then simply X for the FeSCN2 plus concentration. And then remember, FeSCN2 plus concentration is what we get from this is equilibrium, this is what we get from the absorbance. So you'll have the absorbance for this solution, you'll use the calibration graph to determine the concentration, and that means you know this X value, so you'll know it here and you'll know it here. You will have calculated the initial molarities from the dilution, and so that means we can now plug these into the equilibrium constant expression, K sub C, and we'll plug in whatever we got for X, whatever we got for the Fe3 plus, some number minus X, and the SCN minus, some number minus X. That will yield a number. You'll do this same exact thing for all, I believe it's 5 trials that you do. So you'll set up a nice table, use the absorbance for the FeSCN2 plus for that particular trial, grind it all through, calculate your K sub C value. It should be pretty similar to the ones you get for all the other trials. Make an average. Recall that we need to report not just the equation and the K sub C value, but also the temperature at which this happens. And that is it. So that will be your conclusion for this reaction. The K sub C value is whatever you find at 25 degrees Celsius. Okay, so I hope this is a helpful screencast in getting you through the lab. If you have questions, that can be addressed during lab class meetings as well.